The whispers of life's instruction book. Before we drift into the vastness of the cosmos, let's first turn our gaze inward to the quiet hum within you. Imagine a hidden language written in whispers, a blueprint for every verve, every thought, every beat of your heart. This is the story of life's own instruction book unfolding softly. Close your eyes now, if you haven't already, and feel the gentle rhythm of your breathing. A soft, steady inhale, a slow release. With each breath, feel yourself sinking deeper effortlessly into the comfort of your space. The world outside begins to fade, its sharp edges softening its noises, growing distant like the faint echoes of a dream. Here in this quiet stillness, we begin our journey. Tonight, our exploration isn't of distant stars or swirling galaxies, but of a universe far more intimate, far more wondrous, the universe within you. It's a universe teeming with activity even as you lie perfectly still. A symphony of tiny, intricate processes happening at every moment, orchestrated by something remarkable. Imagine for a moment that your body is a magnificent ancient library. Not a dusty, silent place, but one alive with whispers, with the rustle of turning pages, with the quiet hum of countless stories. Unfolding in this library, every cell, every tissue, every organ is a meticulously crafted book. And within each of these books, there are even more delicate scrolls filled with instructions, with recipes, with the very essence of what makes you you. These scrolls, these invisible blueprints, are written in a language far older than any human tongue, Ao. Language composed not of words and letters, but of four gentle symbols repeated and arranged in endless elegant sequences. This is the language of DNA, the very fabric of life's instruction book. It's a language so fundamental, so universal, that it speaks not just in your cells, but in the leaves of a tree, in the fur of a sleeping cat, in the quiet depths of the ocean, where ancient creatures drift. Think of your DNA as a magnificent, impossibly long story. A story that began not with your birth, but billions of years ago, with the very first spark of life on Earth. It's a story passed down through countless generations, each ancestor adding a tiny, unique chapter, refining the narrative, adapting it to new environments, new challenges. And now that story resides within you, a living, breathing testament to an unbroken lineage. Each of your cells, from the tip of your smallest toe to the softest strand of your hair, holds a complete copy of this story. It's like a tiny, perfect microcosm of the entire library, contained within a space too small to see with the naked eye. And within this tiny space, the story is constantly being read, interpreted, and acted upon. It's the silent director guiding the production of every protein, every enzyme, every structure that allows you to breathe, to think, to feel. Consider the gentle curve of your finger or the unique color of your eyes. These seemingly simple traits are not accidental. They are carefully described in the vast, detailed chapters of your DNA. It's a precision so profound, so intricate that it's almost beyond our grasp, yet it unfolds effortlessly, continuously within you. And within this vast narrative, there are specific passages, distinct paragraphs we call genes. Each gene is like a tiny self-contained story carrying instructions for a particular task. Perhaps one gene holds the recipe for making the hemoglobin that carries oxygen in your blood, allowing you to breathe deeply and peacefully. Another might guide the development of the delicate photoreceptor cells in your eyes, letting you perceive the world in all its subtle beauty. These genes are not always active, not always being read. Some are like quiet chapters waiting for the right moment, the right signal to awaken and begin their work. Others are constantly active, humming with purpose, ensuring the smooth functioning of your body even as you slip into slumber. It's a dynamic, ever-changing library, constantly adapting, constantly responding to the subtle cues of your environment. Imagine this intricate book of life nestled deep within each cell, spiraling gently like a double helix staircase, stretching upwards, seemingly without end. Each step on this invisible staircase is a pairing of those four gentle symbols, always in their perfect complementary dance, adenine always whispering to 𝗧𝗵𝗲, always finding its partner in cytosine. This precise pairing is the secret to its remarkable stability, its ability to store and transmit an astounding amount of information across vast stretches of time. This isn't a static blueprint, however, life by its very nature is fluid, adaptable. And sometimes in the quiet unfolding of this grand story, a tiny whisper might change, a single letter might be misplaced. Most often, your body has remarkable ways of correcting these small misprints, of mending the story back to its intended form. But sometimes a change persists, a new variation arises, and this is how life evolves, how species adapt, how the grand narrative of existence continues to unfold in ever new directions. So as you rest, know that within you, a magnificent story is being told. A library of life is quietly humming. It is a story of incredible complexity, of ancient wisdom, and of an ongoing dialogue with the world around you and in the quiet corners of this immense library, in the very heart of this ancient language lie secrets that science has only just begun to uncover. Secrets about how life writes and rewrites its own destiny, and how perhaps we might one day learn to gently guide its hand. In the quiet depths of this cellular world, a remarkable discovery has brought us closer than ever to understanding how life's story is truly written, and how in ancient times, tiny beings learned to edit their own survival. Chapter 2 echoes from the ancient world. Bacteria's secret, long before us in the deep quiet of the primordial oceans, tiny beings held a secret, a defense whispered through generations. Imagine them facing invisible threats, learning to remember, to adapt, to gently defend their existence. This ancient wisdom holds a key to life's intricate dance. As you continue to drift deeper into comfort, let your mind travel back, far, far back in time. Not just centuries or millennia, but billions of years. Imagine a world still young, a planet where land was barren, and life, if it existed at all, pulsed quietly in the warm primordial oceans. In those vast ancient waters, life began simply. Tiny single-celled organisms, invisible to our eyes, were the pioneers. They were the ancestors of everything we see today. The trees, the birds, the fish, and yes, even you. These microscopic beings, the bacteria, were masters of survival. They didn't have complex brains or intricate organs, but they possessed an incredible inherent wisdom encoded within their own simple DNA. Their world was one of constant struggle, a silent, invisible battle for existence, and among their most persistent adversaries were even tinier entities, the viruses. Imagine these viruses as microscopic invaders, like unseen phantoms drifting through the ancient ocean currents. They weren't truly alive in the way a bacterium was, but they were cunning. Their sole purpose was to hijack a living cell, to insert their own genetic instructions, and to force that cell to become a factory for more viruses. For a bacterium, a viral infection was often a death sentence. It was a threat that could wipe out an entire community, a challenge to their very survival. But over eons, in the face of this relentless assault, the bacteria developed a secret, a whisper of defense refined and passed down through countless generations. It was a memory system, an adaptive shield that allowed them to recognize and neutralize these viral threats. Think of it like this: if a bacterium survived a viral attack, it somehow learned to remember that particular invader, and not only remember it, but to pass that memory on to its offspring. This ancient biological memory system is something truly profound. Imagine a small, humble creature without eyes or ears, yet capable of learning from experience and sharing that lesson. It's as if they had a tiny, invisible scrapbook where they kept snippets, mugshots of their viral enemies. When a virus attacked, the bacterium would try to capture a piece of its genetic material, a unique identifier, and carefully store it away. Where did they store these precious memories? Right there within their own DNA, within that long spiraling story of their existence. But they didn't just haphazardly stick them anywhere. They integrated these viral snippets into specific organized regions of their own genome. These regions were characterized by repeating sequences of DNA, like a recurring chorus in a long scientific song. And in between these repeating choruses, like unique verses, were the stored viral memories. This unusual arrangement of repeating DNA sequences interspersed with unique spacer sequences derived from invading viruses is what scientists would much later call CRISPR. The name itself sounds a bit like the crisp, clear sound of breaking something, or perhaps the precise cut of a sharp tool, but for now let's just hold on to the image of these repeating echoes in the bacterial genome holding vital information. So when a bacterium encountered a virus it had seen before, or a descendant of a virus its ancestors had encountered something remarkable happened, that stored memory, that viral snippet in its CRISPR array would be activated. It was like pulling out the mugshot from the scrapbook, but how would the bacterium use this memory to defend itself? This is where the secret truly deepened. The bacteria also evolved special helper molecules like loyal guardians or molecular bodyguards. These guardians were enzymes, proteins, capable of cutting and manipulating DNA. Scientists would later name some of them Cas proteins, short for CRISPR-associated proteins. Imagine them as tiny, highly skilled molecular security guards waiting for instructions. When a bacterium recognized an invading virus through its CRISPR memory, it would then activate these Cas proteins. The Cas protein would use the stored viral snippet as a guide, like a treasure map, leading to the invader's own genetic material. It would precisely seek out the matching sequence in the invading virus's DNA, and once found with incredible precision, the Cas protein would snip like a tiny pair of molecular scissors right through the viral DNA, disabling it. It would effectively disarm the enemy, stopping the infection in its tracks. This was, and still is, a brilliant and elegant defense mechanism. It allowed bacteria to develop an adaptive immunity, a way to learn from past infections and protect future generations. It's a natural ancient system of genetic editing honed over billions of years in the quiet competitive crucible of life. It's a testament to the ingenuity of evolution, the way life finds solutions to its most profound challenges. This quiet, unassuming system, perfected in the smallest, simplest forms of life, lay hidden for eons, a silent whisper in the genetic code. No one knew its true significance or the power it held until much later. It was a secret kept by bacteria, a silent promise of what was possible, waiting patiently for human curiosity to finally catch its faint ancient echo. And it was these very echoes, these mysterious repeating patterns in bacterial DNA, that would eventually capture the attention of curious minds, setting the stage for one of the most profound scientific discoveries of our time. Chapter 3. The Discovery, a Gentle Glimmer in the lab. In the quiet hum of a laboratory, under soft lights, curiosity, like a gentle breeze, stirred a new understanding. Imagine scientists patiently observing, unveiling a system so elegant, so precise, hidden in plain sight. A glimmer of insight, promising profound possibilities, began to emerge as the echoes of ancient bacteria fade softly. Let's gently fast forward, sweeping through billions of years, past the rise and fall of civilizations, past the quiet blossoming of countless life forms. Our journey now brings us to a more recent time, to the quiet, methodical world of human laboratories, places where curiosity is the guiding star and patience is the most valuable tool. Imagine a laboratory in the stillness of night, or the soft glow of morning, filled with the hushed hum of machines and the subtle scent of sterile cleanliness. Here, under gentle lights, dedicated scientists were not searching for grand solutions to human illness or profound genetic mysteries. No, their initial quest was far more subtle, seemingly unassuming. They were simply trying to understand bacteria, One of these curious minds, a Japanese scientist named Yoshimi Ishino, was working with a common bacterium called E. coli in the late 1980s. He was studying how these tiny organisms use an enzyme to transport certain molecules. As he meticulously analyzed the bacterium's DNA sequence, he stumbled upon something peculiar, something that didn't immediately make sense. He found a series of short, repeating DNA sequences separated by unique, non-repeating segments. It was like finding a strange rhythmic pattern in a long, otherwise straightforward book. He published his findings, but the true significance of these odd repeats remained a mystery, a gentle puzzle waiting for the right moment to be solved. Years passed. Other scientists working independently in different parts of the world began to notice similar patterns in the DNA of other bacteria and archaea, another type of ancient single-celled organism. It was as if these repeating whispers were a universal language among these tiny life forms, a secret code hidden in plain sight, yet no one knew what it meant. Then in the early 2000s, a Spanish scientist named Francisco Mojica was studying archaea that thrive in extremely salty environments. He observed these same mysterious repeating sequences. What set his work apart was his keen eye for detail and his persistent curiosity. He noticed something truly remarkable about the unique spacer DNA sequences nestled between the repeats. He realized that many of these spacer sequences were exact matches to DNA from viruses that specifically attacked bacteria and archaea. Think of the thrill of that moment, a quiet spark of insight. In the hum of the lab, it was like discovering that the strange blank pages in a book were actually filled with tiny, coded messages, and those messages were mugshots of old enemies. This was a profound connection. Mojica theorized that these repeating patterns with their embedded viral memories were part of an adaptive immune system, a way for bacteria to remember and defend against viral attacks. He even coined the term CRISPR, which stands for Clustered Regularly Interspaced Short Palindromic Repeats. It's a mouthful, but it simply describes those rhythmic repeating patterns with the viral spacers in between. Mojica's hypothesis was revolutionary, yet it still needed deeper understanding. How exactly did this system work? How did the bacteria use these stored memories to fight off invaders? The mechanism remained elusive, a beautiful puzzle waiting for its final pieces. This is where the story takes another fascinating turn, leading us to two brilliant minds working on different continents, who would ultimately unravel the molecular mechanism of CRISPR. One of these scientists was Jennifer Doudna, a biochemist at the University of California, Berkeley. She had a deep fascination with RNA, a molecule similar to DNA but with distinct roles. In the cell, think of RNA as a temporary message, a working copy of a DNA blueprint carrying instructions from the nucleus to the cells. Protein-making machinery. Doudna was studying how RNA molecules could guide proteins in various biological processes. Across the ocean in Sweden, Emil Charpentier, a microbiologist, was studying 𝗧𝗵𝗲𝗄𝗏 𝗪𝗔, a common bacterium known for causing strep throat. She was particularly interested in how this bacterium defended itself against viruses. She discovered a crucial previously unknown RNA molecule within the CRISPR system of 𝗧𝗵𝗲𝗄𝗏 𝗪𝗂𝗪. She named it tracRNA. This tracRNA seemed to play a vital role, acting like a scaffold or an adapter. Recognizing the potential synergy of their independent discoveries, Doudna and Charpentier decided to collaborate. Imagine them, two brilliant minds from different specialties coming together, driven by shared curiosity. Their collaboration was like two separate streams converging to form a powerful river, each bringing unique insights to the flow of discovery. Their groundbreaking research focused on a specific Cas protein, one of those molecular security guards we mentioned earlier, called Cas9. They hypothesized that Cas9 was the enzyme responsible for cutting DNA, that they needed to understand how it was guided. Their elegant experiments revealed something truly extraordinary. They showed that the CRISPR array with its stored viral snippets produced a special guide RNA. This guide RNA was like a precise search and find mission. It would bind to the Cas9 protein, forming a molecular complex. The guide RNA with its embedded viral memory would then lead the Cas9 enzyme directly to any matching viral DNA a sequence it encountered. Once the match was made, the Cas9 protein, acting as those molecular scissors, would precisely snip both strands of the viral DNA, effectively neutralizing the threat. This was the aha moment, the gentle glimmer that illuminated the entire system. It showed with stunning clarity how bacteria had developed an incredibly precise and adaptable immune system. But the true genius of their discovery lay in its simplicity and its profound implication. If this bacterial system could be harnessed, if it could be programmed with any guide RNA, then perhaps we could direct these molecular scissors to any specific DNA sequence we chose in any organism. The potential was staggering. Imagine being able to edit life's instruction book with such precision, not just in bacteria, but in human cells and plants and animals. It was like discovering a universal word processor for DNA, an ability to read, to find, and to gently edit the very sentences of life. This glimmer of insight born from quiet observation and meticulous experimentation promised profound possibilities that extended far beyond the ancient world of bacteria. It began to paint a picture of a future where we might rewrite life's code for a new, healthier tomorrow. And yet, as the dust settled from this incredible discovery, another layer of questions began to form, questions about the journey from these tiny bacterial secrets to the vast potential of applying this precision in our own complex biological worlds. Chapter 4, The Molecular Scissors. Precision in the microcosm. Imagine tiny, intricate tools, smaller than you can conceive, moving with incredible grace, guided by a perfect plan. These are the molecular scissors designed by nature itself, capable of finding a single word in a vast library and gently, precisely making a quiet change. As the quiet hum of scientific discovery settles around you, let your thoughts now focus on the incredible tools themselves. We've spoken of a language, a book of life, and an ancient memory system. Now let's marvel at the instruments that make the editing possible, the molecular scissors. These aren't scissors you hold in your hand, or even those you see under a powerful microscope. They're far, far smaller, existing in a realm that defies easy visualization, yet their work is profoundly impactful. Picture something so tiny it exists within the liquid world inside a single cell. Something that navigates the intricate, bustling city of proteins and molecules with unparalleled precision. These are the Cas proteins, and specifically the one that has become most famous for its remarkable ability. Cas9. Imagine Cas9 as a highly skilled artisan, a sculptor working on the most delicate of materials. Its primary purpose, its inherent nature, is to interact with DNA, but it's not a random interaction. Cas9 is designed for a specific task: to cut the DNA helix, to make a precise break in that long spiraling staircase of genetic information. To understand its precision, think of your DNA as a vast ancient scroll, thousands of pages long, filled with countless words and sentences. If you wanted to find a specific word on a specific page in a specific line, you'd need an incredible guide, wouldn't you? Cas9 on its own is just a pair of scissors. It needs a map, a detailed instruction set to know where to cut. This map comes in the form of a special molecule we briefly touched upon, the guide RNA. Remember RNA as the temporary messenger, the working copy of DNA. In the world of CRISPR, this guide RNA is truly extraordinary. It's like a custom-made key designed to unlock a very specific door. It's relatively short, just a small string of those 4 basic genetic symbols, but its sequence is crucial. It's been engineered, or rather evolved by nature, to perfectly match a particular sequence of DNA that needs to be found. So picture this: the Cas9 protein, the molecular scissors doesn't operate alone. It forms a partnership, a close embrace with its specific guide RNA. Together, they become a search and destroy team. The guide RNA, with its unique sequence, acts like a homing beacon. It constantly scans the vast stretches of DNA inside a cell, unwinding tiny sections of the double helix, gently probing, searching for its perfect match. Imagine the guide RNA as a tiny magnet specifically attuned to one particular type of metal. It glides along the DNA strand, passing countless other sequences until it encounters the exact pattern of genetic letters it's designed to recognize. This recognition is based on perfect complementary pairing, where the guide RNA has an A, the DNA it seeks must have a T, where it has a G, the DNA must have a C, and so on. It's a lock and key mechanism, utterly precise. When this perfect match is made, when the guide RNA finds its target sequence within the DNA, a transformation occurs. The Cas9 protein, which was once just a dormant pair of scissors, springs to life. The guide RNA has successfully led it to the correct address, the exact line, the precise word in the vast book of life, and then with exquisite precision, the Cas9 enzyme performs its delicate work. It makes a cut, a double-stranded break right through the DNA helix. It's not a rough tear, but a clean molecular incision. Think of it like cutting a single thread in a woven tapestry without disturbing the threads around it. This is the act of the molecular scissors, this ability to make a precise cut at a predetermined location in the DNA is what makes CRISPR-Cas9 so revolutionary. Before this discovery, scientists could make changes to DNA, but it was often a far less precise process, like trying to edit a book by dropping a bomb on a paragraph. You might get the change you want, but you'd also cause a lot of unintended damage around it. CRISPR, in contrast, offers an unprecedented level of control, like using a surgeon's scalpel, where before you had only a blunt instrument. Why is making a cut so important? Because a cell's natural response to a break in its DNA is to try and repair it. Your cells are constantly performing maintenance, patching up damage, mending broken strands, and this natural repair machinery can be subtly guided. When Cas9 makes its cut, the cell scrambles to fix the break. It has two main pathways for this repair. One pathway is a bit like a quick emergency patch-up job. It's called non-homologous end joining, or NHEJ. This pathway tends to be a bit messy. It might add a few extra genetic letters or delete a few when it glues the DNA back together. These small additions or deletions can disrupt a gene, essentially turning it off. Imagine you cut out a sentence from a book, and when you glue the pages back together, a few words are missing or added, making the sentence unintelligible. This can be incredibly useful if you want to silence a faulty gene to make it stop producing a harmful protein. The second repair pathway is far more precise. It's called homology-directed repair, or HDR. This pathway requires a template, a perfectly matching piece of DNA to guide the repair. Imagine cutting a sentence out of a book, but then you immediately provide the cell with a perfect corrected version of that sentence. The cell's repair machinery can then use your provided template to meticulously fill in the gap, inserting the new desired genetic sequence. This is the pathway that allows for truly targeted genetic correction for rewriting a specific word or sentence in life's instruction book. The elegance of this system, its simplicity and precision, is what makes it so powerful. It's a biological marvel refined over billions of years within the smallest of organisms, now understood and potentially harnessed by human ingenuity. The molecular scissors, guided by their intricate RNA maps, offer a quiet promise: the ability to edit life's code with a clarity and control we could only dream of before. This newfound ability to precisely cut and guide the repair of DNA opens up vast possibilities. It gently leads us to consider how we might use this incredible precision its quiet power to mend the stories within ourselves and perhaps even in the wider world around. US Chapter 5, Guiding the Scissors, a map for the unseen. But how do these tiny tools know where to go, what to snip? Imagine a delicate map, a whispered instruction guiding them through the labyrinth of life's code. This invisible guidance system ensures precision, like a dream leading you to a forgotten memory. As the image of the molecular scissors, so precise and powerful, gently fades, let your mind now settle on a different aspect of this wonder. A pair of scissors, however, sharp is useless without a hand to guide it, without an eye to see the line to be cut in the microcosm of the cell. Who or what provides this intricate guidance? How? Do these tiny tools know exactly where to go, what specific sequence of genetic letters to snip amidst the vast sprawling expanse of your DNA? This is where the magic of the guide RNA truly shines. Imagine your entire genome, the full instruction book of your life as a library so immense it contains billions of words. If you wanted to find one single specific word, perhaps a unique misspelling amidst all those billions, how would you do it? You'd need an incredibly precise search function, a map that leads you directly to that exact spot. The guide RNA is exactly this map, this whispered instruction for the unseen. It's a remarkable molecule, an ephemeral whisper of genetic code crafted by scientists to mimic the natural system found in bacteria. Remember that RNA is like a temporary working copy of DNA, but this guide RNA is special. It's designed with a very specific purpose. Think of it as a custom-made key. Each key has a unique pattern of teeth designed to fit only one specific lock, and the same way each guide RNA has a unique sequence of its own genetic letters, as USRNA's equivalent of DNA's T. C, S, and Gs that is precisely complementary to a specific target sequence in the DNA you wish to alter. So how does this map work? In practice first, the Cas9 protein, our molecular scissors, isn't just floating around aimlessly. It forms an intimate partnership with this guide RNA. They bind together, becoming a single functional unit, like a diver preparing with their oxygen. Tank and flippers. The guide RNA essentially slips into a special groove within the Cas9 protein, becoming its eyes and its compass. Once this partnership is formed, the guide RNA begins its search. Imagine it gliding along the long winding double helix of your DNA, like a gentle explorer navigating an endless river. As it moves, it continuously reads the genetic landscape. It does this by gently unzipping small sections of the DNA, double helix, just enough to expose the hidden letters within. With each tiny stretch of unzipped DNA, the guide RNA attempts to form temporary bonds with the exposed genetic letters. It's like testing a series of keys in a row of locks. It's looking for its perfect match, its complementary sequence. If the guide RNA has an A, it's looking for a T in the DNA. If it has a G, it's seeking a C. This process of searching and matching is incredibly fast and efficient. The guide RNA will ignore vast stretches of DNA that don't match its sequence. It's not looking for anything similar. It's looking for an exact fit, like finding the one true echo of its own whisper. When the guide RNA encounters a DNA sequence that is perfectly complementary to its own, a powerful recognition occurs. It's like the key finally sliding into its lock, clicking satisfyingly into place. This precise pairing is the signal. This is the moment the guide RNA has found its target, the specific word or sentence in the vast genetic library that you wish to interact with, crucially theirs. A tiny additional signal that ensures even greater precision. Right next to the target sequence that the guide RNA recognizes, there's usually a short specific DNA sequence known as the PAM sequence. Protospacer adjacent motif. Think of the PAM sequence as a special punctuation mark, like a period or an exclamation point that tells the Cas9 system you are at the right address and this is the specific spot to cut. Without the presence of this PAM sequence, the Cas9 enzyme will not activate its cutting function, even if the guide RNA finds a matching sequence. It's an extra layer of security, a safeguard against unintended cuts, ensuring that the molecular scissors only operate at very specific predetermined locations. Once the guide RNA has locked onto its target with the help of the PAM sequence, the Cas9 protein, which was merely a passenger until now, undergoes a subtle but significant change in its shape. This conformational shift activates its cutting ability, and then with that incredible precision we spoke of, the Cas9 makes its double-stranded break right at the exact location designated by the guide RNA. This entire process, from the initial search to the final cut, happens within the microscopic confines of a cell, a place of constant motion and chemical reactions, yet it unfolds with remarkable accuracy. Billions of times over in bacterial defense, and now with astonishing control in human-engineered systems. The beauty of this guidance system lies in its modularity, because the guide RNA is separate from the Cas9 protein, and because its sequence dictates, the target scientists can simply design a new guide RNA for almost any DNA sequence they wish to target. This means that with the same Cas9 scissors, you can effectively cut at countless different locations simply by swapping out the map. It's like having a universal cutting tool that can be programmed for endless different tasks just by changing its input instructions. This invisible guidance system, refined over eons in the ancient world of bacteria, provides the exquisite control necessary for gene editing. It ensures that the molecular scissors don't just snip randomly, but rather with the focused intent of a dream leading you to a forgotten memory. It is this profound ability to guide the molecular scissors that unlocks the true potential of CRISPR, leading us to contemplate how we might use this precision to mend and rewrite life's stories, gently correcting paths that have gone astray. Chapter 6, Rewriting the Story, Mending and Mending Again. With these gentle tools and their quiet guidance, imagine the possibility of rewriting a single sentence, of mending a story that has gone astray. It's not about erasing, but about softly correcting, allowing life's narrative to unfold more harmoniously as the image of the invisible guide RNA, leading its molecular scissors, gently fades. Let your mind now rest upon the profound implications of this precision. We've journeyed deep into the microscopic world, glimpsing the ancient defenses of bacteria and the ingenious tools that nature has perfected. Now we arrive at a truly awe-inspiring concept, the ability to rewrite the story of life itself. Imagine once more your DNA as a vast, incredibly long book, the ultimate instruction manual for you. Sometimes in the billions of letters that make up this book, a tiny typo occurs. A single letter might be swapped for another, or perhaps a sentence might be accidentally deleted, or an extra nonsensical word inserted. These small typos, or mutations as we call them in the language of science, are often harmless. Your body has remarkable ways of dealing with them, often simply ignoring them or repairing them, but sometimes, just sometimes, a particular typo lands in a critical part of the story, in a vital sentence or a crucial paragraph, a gene. When this happens, the instructions become garbled. The cell might no longer be able to make a necessary protein, or it might produce a faulty one. This is often the root cause of many inherited diseases, conditions that are passed down through generations silently, written into the very fabric of one's being. Think of a story where a single vital word is misspelled, changing the entire meaning of a sentence, leading the plot astray. For example, a gene that instructs your body to make a protein for healthy vision might have a tiny error leading to a condition, like certain forms of blindness, or a gene responsible for making hemoglobin, the molecule that carries oxygen in your blood, might be altered, leading to conditions like sickle cell anemia. For a long time, correcting these typos was a dream, a whisper. On the wane of scientific aspiration, it was like wanting to edit a single word in a book, that was already printed, bound, and distributed to billions of people. It seemed almost impossible, but now with the gentle precision of CRISPR, that dream begins to take shape with the Cas9 molecular scissors guided by its custom-made RNA. MAP scientists can now aim with unprecedented accuracy at these specific genetic typos. It's not about erasing entire chapters or rewriting whole paragraphs at random. It's about softly correcting, about targeting a single precise spot in that vast genetic library. When the Cas9 guide RNA complex finds its target, that tiny faulty sequence in the DNA, it makes its precise cut. Remember, the cell's natural response to a DNA break is to repair it, and this is where the rewriting truly begins, through the cell's own innate mending processes. One way the cell repairs the cut is through that emergency patch-up pathway we touched upon, called non-homologous end joining. Hey, this pathway is a bit like quickly taping two torn pieces of paper together. It's efficient, but it can be a bit messy. When the cell tries to glue the DNA back together using NHEJ, it might accidentally add a few extra genetic letters or delete a few. These small additions or deletions can act like a genetic off switch if they occur within a gene. They can introduce errors that completely disrupt the gene's ability to produce a functional protein. Why would we want to turn a gene off? Imagine a disease where a faulty gene is producing a toxic protein or a protein that is causing harm. In such cases, simply turning that gene off, silencing its harmful message, could be a therapeutic approach. It's like silencing a noisy, out-of-tune instrument in an orchestra to restore harmony. But the true power, the most elegant form of rewriting, comes from another natural repair pathway: homology-directed repair, HDR. This pathway is far more refined. It's like repairing a torn page by carefully stitching it back together using a perfect template as a guide. When the Cas9 creates a break in the DNA, scientists can introduce a new corrected piece of DNA into the cell. This new piece acts as the template. Imagine that you have the original pristine version of the sentence that was misspelled in the book of DNA. When the cell's repair machinery sees the break, it looks for a template to guide the repair, and there, conveniently, is the perfect corrected version you've provided. The cell then uses this template to meticulously patch up the broken DNA, inserting the new correct sequence. This is the essence of gene correction, or gene editing, in its most precise form. It's not about adding entirely new genes willy-nilly, but about correcting existing ones. It's about changing an A to a G where a G should have been, or inserting a missing sequence that is crucial for a gene to function properly. It's about mending the story line by line, allowing the narrative of life to unfold as it was meant to, or perhaps even more harmoniously than before. Think of the implications for diseases like cystic fibrosis, where a single tiny mutation prevents a crucial protein from working correctly, leading to thick mucus buildup in the lungs and other organs. With CRISPR, one day it might be possible to precisely correct that specific mutation in the affected cells. Or for Huntington's disease, a devastating neurological disorder caused by an abnormal expansion of a repetitive DNA sequence, CRISPR might offer a way to excise or silence that problematic expansion. This ability to gently correct the genetic code is not just about fixing what is broken. It also opens doors to subtly enhancing existing traits, or even inserting entirely new beneficial ones, perhaps making crops more resilient to drought or preventing certain infections in animals. The possibilities, while still in their early stages of exploration, are vast and profound. It's a truly humbling thought that the precise, elegant defense mechanism evolved by ancient bacteria, honed over billions of years to protect themselves from viruses, could now be harnessed by human hands to mend the very fabric of our own existence. It is a testament to the interconnectedness of all life, a silent whisper from the distant past, now empowering us to softly rewrite the future. this silent revolution born from understanding how life mends itself, gently pushes us toward a new dawn where healing might come, not just from outside, but from the deepest, most fundamental level within us. Chapter 7, A New Dawn for Health, Healing from Within. Now let your mind drift to the idea of profound healing, not from outside, but from deep within your own cells. Imagine a future where whispers of illness can be quieted, where the body's own wisdom is gently guided back to balance. A new dawn, perhaps, for health and well-being. As you continue to settle into this peaceful space with the soft images of genetic mending still lingering, let your thoughts gently expand to embrace a profound, hopeful possibility. For centuries, when illness struck, humanity has sought cures from external sources. Medicine, surgeries, therapies applied from the outside in. But what if healing could begin not just with a pill or a procedure, but from the very deepest, most fundamental level of your being? What if we could quiet the whispers of illness, not by silencing symptoms, but by gently correcting the faulty instructions at their source, deep within your own cells? This is the promise that CRISPR, this remarkable molecular system, brings to the dawn of health. Imagine a future where the story of your life, if it were to veer off course due to a genetic misprint, could be softly guided back onto a path of wellness, not through invasive interventions, but through precise, almost imperceptible adjustments within your own body. Consider the landscape of human disease. Many conditions, from rare inherited disorders to more common ailments like certain cancers or infections, have a genetic component. This means that somewhere in that vast book of your DNA, there's a particular typo, a misplaced letter, a missing word, or an extra unwanted sentence that contributes to the illness. For decades, these genetic misprints felt like an unchangeable fate, like immutable sentences written in stone. But now, with the understanding of how to guide those molecular scissors, the Cas9 protein, to a precise location, and how to encourage the cell's natural repair mechanisms, a new chapter in medicine is slowly beginning to unfold. One of the most immediate and impactful applications of CRISPR lies in treating inherited genetic disorders. Think of diseases like cystic fibrosis, where a single mutation in a gene leads to the production of abnormally thick mucus, clogging the lungs and other organs. Or sickle cell anemia, where a single change in the gene for hemoglobin causes red blood cells to deform, leading to chronic pain and organ damage. For these conditions, CRISPR offers a precise scalpel to correct the specific genetic error. Imagine taking a patient's own cells, perhaps from their bone marrow, correcting the faulty gene using CRISPR, and then reintroducing those healthy corrected cells back into their body. This approach, known as ex vivo gene editing, is already showing remarkable promise in early clinical trials for sickle cell disease and beta thalassemia, another blood disorder. Patients who once faced lifelong transfusions or debilitating pain are beginning to experience profound improvements, their bodies starting to produce healthy blood cells once more. Another area where CRISPR shines a hopeful light is in the fight against cancer. Cancer at its core is a disease of uncontrolled cell growth, often driven by accumulated genetic mutations. CRISPR could be used in several ways here. Imagine enhancing your own body's immune cells, called T cells, to make them far more effective at recognizing and destroying cancer cells. Scientists are using CRISPR to edit cells, arming them with better targeting mechanisms or removing internal breaks that prevent them from fully attacking tumors. These genetically enhanced T cells can then be infused back into the patient, unleashing a potent, personalized internal army against the cancer. This approach is part of a rapidly advancing field called cell therapy, and CRISPR is making it even more precise and powerful. Beyond cancer, consider chronic viral infections. Like HIV, the HIV virus integrates its own genetic material into the host's DNA, making it incredibly difficult to eradicate. CRISPR offers the tantalizing possibility of precisely cutting out the integrated viral DNA from infected cells potentially, leading to a functional cure. While still in early research phases, the idea of literally excising a persistent viral intruder from your genetic story is a truly revolutionary concept. And it doesn't stop there. The whispers of CRISPR's potential extend to neurological disorders like Huntington's disease, where a toxic protein is produced due to a genetic expansion. CRISPR could potentially be used to silence the problematic gene or even to shorten the expanded sequence. For conditions like Duchenne muscular dystrophy, where a large gene has a small but critical missing piece, CRISPR might be able to snip out the faulty section and encourage the cell to create a shorter but functional version of the protein. The beauty of CRISPR lies in its versatility. It's not a one-size-fits-all drug, it's a foundational technology that can be adapted to countless different genetic targets. It operates on the principle that the cell itself has the ultimate power to heal given the right instructions. CRISPR simply provides those instructions, acting as a gentle guide for the cell's own repair machinery. Imagine the profound implications for personalized medicine. Each person's genetic story is unique, and so are their genetic susceptibilities. With CRISPR, it might one day be possible to tailor treatments precisely to an individual's unique genetic makeup, addressing their specific typos with unparalleled accuracy. This moves us beyond broad treatments to highly targeted, individualized therapies truly healing from within. However, it is important to remember that this new dawn is still unfolding. While the promise is immense, the journey from laboratory discovery to widespread clinical application is long and complex. There are still challenges to overcome, ensuring that the CRISPR components get to the right cells in the body delivery, making sure they act with absolute precision without making unintended cuts elsewhere, off-target effects, and understanding the long-term consequences of these genetic changes. Yet the speed of progress has been breathtaking. What began as an ancient bacterial defense mechanism, then the scientific curiosity, has very quickly transformed into a powerful tool with the potential to fundamentally redefine our approach to health and disease. It is a testament to the quiet power of curiosity, leading us from the smallest echoes of life to the grandest visions of human well-being. This ability to mend the intricate threads of life, to correct the quiet errors that cause so much suffering, draws us into a thoughtful consideration of our responsibility as we learn to wield such profound power over the very essence of life. Chapter 8, The Ethical Embrace, Navigating the Unknown. As we explore these gentle possibilities, a quiet question arises, a soft whisper of responsibility in the vastness of discovery. Imagine balancing wonder with wisdom, considering the delicate threads that connect us all. This journey requires not just knowledge, but thoughtful reflection. As the hopeful light of healing from within softly illuminates your thoughts, let's gently shift our focus from the how of CRISPR to a deeper, more reflective should, with such profound power to edit the very essence of life, to rewrite parts of that ancient instruction. Book a quiet but insistent question naturally arises: how do we navigate this incredible new frontier with wisdom and responsibility? Imagine standing at the edge of a vast, beautiful, yet uncharted ocean. The horizon shimmers with promise, but the depths hold unknowns. CRISPR, in its powerful elegance, presents us with just such a landscape. It is a technology that allows us to gently touch the very fabric of life, and with that touch comes an immense ethical embrace, a responsibility to consider the delicate threads that connect us all, past, present, and future. One of the most immediate ethical considerations revolves around somatic gene editing. This refers to making genetic changes in the cells of an individual that are not passed on to their children. These changes affect only the treated person. For example, editing blood cells to treat sickle cell anemia or immune cells to fight cancer, as we discussed. Most scientists and ethicists agree that somatic gene editing for the treatment of serious diseases is ethically justifiable, provided it is safe, effective, and undergoes rigorous testing and regulation. It is in essence a new form of therapy akin to a highly advanced medicine. The primary concerns here are ensuring safety, preventing unintended off-target edits, cuts at unintended locations, and ensuring equitable access to these potentially life-changing treatments. However, the ethical landscape becomes far more complex when we consider germline gene editing. This involves making genetic changes to reproductive cells, sperm or eggs, or to early embryos. The critical difference here is that these changes would be inherited by all future generations. Imagine altering a gene in an embryo, and that alteration then becomes a permanent part of that family's lineage, passed down through children, grandchildren, and beyond. This raises deeply profound questions. If we correct a disease-causing mutation in embryo, are we preventing future suffering? Most would agree this sounds like a noble goal, but what if we move beyond correcting disease and begin to consider enhancement? What if we could use CRISPR to engineer embryos for traits like increased intelligence, athletic prowess, or specific physical characteristics? Where do we draw the line between therapy and enhancement? Who decides what constitutes an enhancement, and whose values guide such decisions? Consider the idea of designer babies. While sensationalized, the underlying ethical dilemma is real. If such technologies become widely available, would they exacerbate existing social inequalities? Would only the wealthy have access to these enhancements, creating a new form of genetic aristocracy? Would parents feel pressure to optimize their children, leading to unforeseen societal pressures and expectations? These are not easy questions, and they demand thoughtful, open dialogue involving not just scientists, but ethicists, philosophers, policymakers, and the public. Another quiet concern is the concept of unintended consequences. Life's instruction book, the genome, is incredibly complex, a symphony of interacting genes and regulatory elements. While CRISPR is remarkably precise, making a change in one part of the system could theoretically have unforeseen effects elsewhere, like altering a single note in a grand musical score and subtly changing the harmony of the whole. Though scientists are diligently working to minimize these off-target edits and understand the intricate networks of genes, the full long-term implications of germline changes are by definition unknowable. Until generations have passed, there is also the question of consent. An adult can consent to a somatic gene therapy, but an embryo cannot. Who consents for the unborn, for future generations who will carry these permanent genetic alterations? This forces us to consider our collective responsibility to those who will come after us, to ensure that the choices we make today are truly in their best interests, and do not inadvertently burden them with unforeseen consequences. Furthermore, the very act of manipulating the human germline raises questions about our relationship with nature and the definition of humanity. Some worry about playing God, while others argue that using our knowledge to alleviate suffering is a fundamental human good. There are diverse perspectives across cultures and belief systems, and any global consensus on these matters requires profound respect for differing viewpoints. To navigate this unknown territory, society needs mechanisms for thoughtful reflection and ethical governance. This means encouraging open scientific research while simultaneously fostering public dialogue, establishing clear regulatory frameworks, and perhaps even engaging in international agreements to guide the responsible application of gene editing technologies, especially for germline alterations. Many countries have for now put moratoriums or outright bans on human germline editing for reproduction, reflecting a global caution and a desire for further ethical deliberation and scientific understanding. The journey with CRISPR is not merely a scientific expedition, it is a profound ethical exploration. It asks us to balance the immense wonder of what we can do with the wisdom of what we should do. It compels us to consider the delicate threads that connect all life, not just human life, and to embrace our responsibility as custodians of this powerful knowledge. And as we ponder these profound responsibilities, our thoughts gently turn outward beyond the individual, to consider how this revolutionary ability might shape not just human health, but the very future of life on our planet in ways both subtle and grand. Chapter 9. Beyond ourselves, shaping the future of life, beyond the boundaries of our own bodies. Imagine the quiet ripple effect across all living things, from the tiniest plant to the grandest creature. The whispers of this technology could gently shape futures. A profound connection to the tapestry of life itself, as your mind embraces the ethical considerations of our journey inward. Let us now gently expand our gaze, drifting beyond the confines of the human body, beyond the whispers within your own cells. For the revolutionary precision of CRISPR holds implications far vaster reaching, out to touch the very tapestry of life on Earth, from the simplest bacterium to the majestic forest, from the smallest insect to the largest whale. Imagine the quiet ripple effect across all living things, the same molecular scissors and guiding RNA that hold promise for human healing also possess the capacity to gently shape the future of plants, animals, and even entire ecosystems. This is where CRISPR truly becomes a profound connection to the intricate dance of existence itself. Consider first the world of agriculture and food security. For centuries, humans have engaged in selective breeding, patiently guiding the evolution of crops and livestock to enhance desirable traits, bigger fruits, more resilient grains, healthier animals. This was a slow, sometimes unpredictable process, like choosing threads for a tapestry, one by one without a clear pattern. Now with CRISPR, this process can become far more precise and efficient. Imagine gently editing a gene in a wheat plant to make it inherently resistant to a devastating fungal blight, reducing the need for chemical pesticides and securing vital food supplies for a growing global population. Or perhaps subtly tweaking a gene in a rice plant to make it more tolerant to drought or salty soil, allowing it to thrive in increasingly challenging climates. Think of crops that are more nutritious, capable of producing higher yields with fewer resources, or even engineered to absorb more carbon dioxide from the atmosphere. The implications for a sustainable future are vast. This isn't about creating entirely new species in a fantastical way. It's often about making tiny, targeted adjustments to existing organisms, enhancing their natural resilience and productivity in ways that could otherwise take decades or centuries through traditional breeding. It's like gently fine-tuning a well-designed machine to perform even more efficiently. Beyond plants, consider the potential for animal health and welfare. Imagine protecting livestock from devastating viral diseases by making them genetically resistant, thereby reducing suffering and the widespread use of antibiotics, or perhaps engineering mosquitoes to be unable to carry malaria or dengue fever, potentially saving millions of lives in affected regions. This concept, known as gene drive, aims to spread a desired genetic trait rapidly through a population, offering a powerful, albeit ethically complex tool for controlling disease vectors. It raises careful considerations about ecological impact and reversibility, reminding us again of the need for profound reflection before widespread application. The gentle touch of CRISPR also extends to conservation efforts. Picture the challenge of protecting endangered species from rapidly spreading diseases or from the impacts of climate change. Could CRISPR be used to confer disease resistance to vulnerable populations, or to introduce traits that enhance their ability to adapt to changing environments? Imagine, for instance, making coral more resilient to ocean acidification, or trees more resistant to invasive pests that threaten entire forests. These are delicate and complex undertakings requiring deep ecological understanding, but the tool exists to consider. Such possibilities, this capacity to edit the genetic code of diverse organisms, creates a profound sense of interconnectedness. We are no longer just passive observers of evolution, we are becoming, in a carefully considered way, active participants. In shaping life's ongoing story, this is a humbling realization, one that demands not only scientific rigor, but also a deep reverence for the intricate balance of nature. However, just as with human applications, the use of CRISPR in plants and animals requires careful ethical consideration and robust regulatory oversight. Questions arise about the long-term ecological consequences of introducing genetically altered organisms into the environment. Could unintended side effects occur? Could the engineered traits spread in unforeseen ways? How do we balance the potential benefits of increased food security or disease eradication with the need to preserve biodiversity and ecological stability? The whispers of responsibility here are just as vital. The power to shape life's future is immense, and with it comes the obligation to proceed with caution, with foresight, and with a commitment to understanding the full spectrum of impacts. This means not rushing into widespread applications without thorough testing and public discourse. It means fostering global collaboration to establish guidelines and best practices, ensuring that this powerful technology is used for the good of all and not just for short-term gain. The story of life on Earth is one of continuous change, adaptation, and evolution. CRISPR offers us a new chapter in this story, one where we can perhaps gently guide some of these changes with greater intent and precision. It's a profound connection, a quiet dialogue with the very forces that have shaped life for billions of years. As we learn to understand and utilize these ancient echoes, we are invited to consider our place in the grand tapestry, not just as individuals, but as stewards of this remarkable planet and all its living wonders. And as we contemplate this vast responsibility, this quiet influence we might wield over the future of all life, our journey softly draws us towards its close, inviting us to reflect on the unfolding narrative of discovery itself and the dreams of tomorrow. Inspires Chapter 10. The unfolding story, dreams of tomorrow. As we softly approach the edges of sleep, let your mind wander to the unfolding story, the chapters yet to be written by human ingenuity and care. Imagine a world where understanding deepens and healing blossoms, a gentle promise held within the very fabric of life. Now, as the gentle currents of our journey carry you closer to the tranquil shores of sleep, let your mind drift from the intricate details of molecular scissors and ancient bacterial secrets. Instead, allow yourself to simply imagine, to dream of the unfolding story. We have glimpsed the astonishing power of CRISPR, a power born from nature's own wisdom, now in the hands of human curiosity and ingenuity. What is this profound understanding, this gentle ability to interact with life's code promise for tomorrow? Imagine a world where understanding deepens. The journey of science is never truly finished, each discovery, however monumental, simply opens new pathways for exploration. CRISPR itself continues to evolve. Scientists are constantly refining the molecular scissors, seeking even greater precision, developing new Cas enzymes that might be smaller, more versatile, or capable of editing different types of genetic material like RNA directly. They are exploring ways to deliver these genetic tools to specific cells in the body with unparalleled accuracy, ensuring that the healing touch reaches only where it is needed, like a soft whisper directed to a particular ear. Consider the ongoing quest to understand the complexities of the human genome. Even with the instruction book read, we are still learning how all its billions of letters interact, how genes are turned on and off, how they influence each other in intricate networks. CRISPR becomes an invaluable tool in this quest. By precisely turning genes off or on, or making subtle edits, scientists can meticulously probe the function of each genetic word and sentence, unlocking deeper insights into health and disease. This deepening understanding will in turn inform even more precise and effective ways to use gene editing for healing. Imagine a world where healing truly blossoms not just for a few, but for many. The early clinical trials of CRISPR's therapies are showing remarkable promise, bringing hope to individuals and families burdened by diseases once considered incurable. As these therapies progress, as their safety and efficacy are further validated, we can envision a future where genetic diseases are not just managed, but potentially cured at their root cause. This could mean a world with less suffering, where the genetic predispositions that once whispered threats now whisper promises of wellness. Think of how this technology might intertwine with other medical advancements. Personalized medicine, where treatments are tailored to an individual's unique genetic makeup, could become a profound reality. Diagnostics might become so sophisticated that genetic predispositions to illness are identified even earlier, allowing for preemptive genetic interventions that prevent disease from ever taking hold. Imagine a future where genetic health is simply a part of comprehensive well-being, where the body's own wisdom is truly guided back into balance with gentle, precise adjustments. Beyond direct human therapy, the dreams extend to our planet. Imagine a world where agriculture is more resilient, where crops can feed more people with less environmental impact, adapting gracefully to a changing climate. Imagine animals living healthier lives, less susceptible to devastating diseases. Imagine ecosystems bolstered against threats with endangered species. Offered a helping hand through careful, ethical genetic support. This isn't about imposing our will on nature, but about understanding its profound mechanisms and, where appropriate, subtly assisting its ongoing resilience. Of course, the unfolding story is not without its thoughtful considerations. The ethical questions we gently explored earlier will continue to be vital companions on this journey. The rapid pace of scientific discovery demands an equally rapid and thoughtful evolution of societal dialogue, regulatory frameworks, and global collaboration. We must ensure that this powerful tool is wielded with immense care, with a commitment to equity, access, and foresight, always remembering that the tapestry of life is interconnected and delicate. The dreams of tomorrow are not just about what is scientifically possible, but about what we as a global community collectively choose to build. It's about cultivating a deep sense of responsibility alongside our boundless curiosity. It's about remembering the ancient origins of this technology as simple bacterial defense, and recognizing the profound trust placed in us to use it wisely for the benefit of all life. So, as you feel the gentle post-sleep, let these thoughts linger softly. The story of life written in whispers of DNA is constantly unfolding with CRISPR. We have learned to read some of those whispers with new clarity, and perhaps to add a few lines of our own, guided by ingenuity and care. It's a gentle promise held within the very fabric of life, waiting for the chapters yet to be written by the dreams of tomorrow. The journey of discovery continues even as you rest, leading us ever onward into the vast, quiet potential of our interconnected world.